Hello, my name is Phyllis, and my current research in the JOSTI lab is the development of a nanohoop rotaxane for sensing reactive oxygen species. I am working under the Department of Chemistry and Biochemistry with Ramesh Jasti as my PI and Claire Otteson as my graduate mentor. The JOSTI lab is an organic synthetic lab that specializes in cycloparaphenylenes, abbreviated as Ncpps, where N indicates the number of benzene rings in the structure. CPPs are a new nanostructure with size-dependent fluorescence. As the number of benzene rings decrease, the fluorescence redshifts. The CPP can be utilized in a mechanically interlocked molecule, which is defined as a molecule held together by physical bonds that cannot be separated without breaking a chemical bond between atoms. One benefit that comes out of mechanically interlocked molecules is that the resulting structure possesses highly selective molecular recognition sites that would be difficult to access using traditional covalent bonds. The rotaxane is one type of mechanically interlocked molecule and is composed of a linear component that's threaded through a ring and terminated by bulky end groups or stoppers, large enough to prevent the linear component from slipping out. Our ring of interest is the CPP, which gives rise to the nanohoop-based rotaxane. Because of its modular molecular recognition ability and fluorescence, the nanohoop-based rotaxane is the foundation of a sensing platform that can detect biologically relevant analytes. Preliminary work on the nanohoop-based rotaxanes reveal a turn-on fluorescence response for detection of hydrogen sulfide. In an interlocked state, the structure is non-fluorescent due to the 2,4-dinitrophenol stopper group that acts as a quencher. Hydrogen sulfide chemically reacts with the stopper group, which prompts dethreading of the rotaxane, and this affords a turn-on fluorescence response. Additionally, synthesis of CPP derivatives is well established, including a 2,5 substituted pyridyl embedded CPP shown in the bottom left. This nanohoop is an 8-step synthesis requiring 4 different starting materials and acts as the fluorescent payload for the nanohoop-based rotaxane. Since the 2,4-dinitrophenol stopper group acts as a quencher, it allows for the use of a second stopper as a sensing group for different analytes. This provides a foundation for a modular sensing platform where the sensing component can be interchangeable to react with different biological analytes. My goal is to synthesize a nanohoop-based rotaxane to target reactive oxygen species. Reactive oxygen species exist as free radicals in the body which are unstable and react with other molecules in the cell, making it a biologically relevant analyte that's difficult to study. An imbalance of reactive oxygen species oxidants is connected to severe human diseases such as cancer, cardiovascular disorders, and neurodegenerative diseases. So I began my project by building and optimizing a synthetic scheme for the boronic ester trigger, which is a stopper group that reacts with the reactive oxygen species and prompts dethreading. Starting with starting materials 4-bromobenzaldehyde and 4,5-dinitrobenzaldehyde, I performed a series of reductions and borylations to yield the boronic ester highlighted in red. Next, I synthesized the second stopper group, which is the 2,4-dinitrophenol that acts as the fluorescence quencher and also is the same quencher that they use in the hydrogen sulfide detector. And I've highlighted this in gray. To thread the 2 pieces of the rotaxane into the CPP cavity, an active template method was used. Specifically, the active template CPPs reaction using a copper reagent to catalyze bond formation in the interior of the hoop. The CPP embedded with a 2,6 pyridyl moiety coordinates with the copper reagent to form a bond between the terminal alkyne of the boronic ester trigger and the halo alkyne of the fluorescence quenching trigger. Once the bond is formed, the nanohoop releases the copper catalyst and the linear rotaxane component is mechanically interlocked, hence the name mechanically interlocked molecule. The synthetic portion of my results proved to be a non-trivial process and required weeks of optimization. My future direction include finding more efficient synthetic and purification steps, then characterizing the structure. I will then evaluate the structure under simulated physiological conditions, such as using a sulfonic acid buffering agent with a pH of 7. The expectation is that the structure will be non-fluorescent, but then I will plan to introduce a reactive oxygen species, such as hydrogen peroxide, to the nanohoop rotaxane in both the simulated biological environment and in living cells. Success of the system is indicated by a turn-on fluorescent response to reactive oxygen species. Lastly, I'd like to thank you for listening to my research, and I'd like to acknowledge my research lab, especially Ramesh Jasti and Claire Addison, for making my project possible.